cycle. Interphase. Interphase is the phase of the cell cycle in which a typical cell spends most of its life. During this phase, the cell copies its DNA in preparation for mitosis. Interphase consists of three phases: G1 phase, which is growth, S phase, replication, and G2 phase, preparation for division. First stage of interphase, G1, consists of the cell growing and performing routine activities. During this time, the beginning of protein synthesis occurs and the cells grow to about double its original size. Also, the cell grows and enlarges. Interphase, S phase. S standing for synthesis. At the beginning of the S phase, the cell's nucleus has two pairs of each chromosome, one pair member from each parent. During the S phase, DNA is replicated to produce exactly two identical chromosomes. At the end of the S phase, all of the chromosomes have been replicated. The amount of DNA in the cell has therefore doubled. Interphase G2. At the G2 phase, the cell ensures that it is ready to begin mitosis, occurring between the end of DNA replication in the S phase and the beginning of cell division and mitosis. Also, the cell increases in synthesis of RNA and protein. Also, the G2 phase of interphase is the safest phase of or holding area for the cell. Mitosis. Mitosis is the four-stage process in which identical nucleus are created before cell division. Mitosis is a big part of the cell cycle in which chromosomes in the cell nucleus are separated into two identical sets of chromosomes, and each set ends up in its own nucleus. Mitosis occurs in the animal and plant cells, though they are similar and very different at the same time. Mitosis. Prophase. Prophase is the first phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. Each chromosome is composed of two sister chromatids containing identical genetic information. At the end of prophase, the membrane around the nucleus in the cell dissolves away, releasing the chromosomes. Mitosis, metaphase. Metaphase is the process that separates duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. Metaphase is the second phase of mitosis. The centrioles send out specialized tubules, also known as microtubules. Also, the chromosomes line up and connect the middle and attach spindle fibers. Mitosis, anaphase. Anaphase is the third phase of mitosis. Sister chromatids of each chromosome are pulled apart by the spindle and are dragged by their kinetic toward opposite poles of the cell, that is toward the opposite centrosomes. The spindle fibers shorten the sister chromatids move opposite of the poles of the cell. There are two types of anaphase. Anaphase A, which the microtubules shorten and draw chromosomes toward the spindle fibers. Anaphase B, which is microtubules that are anchored to the cell membrane, pull the poles further apart, and the interpolar microtubules slide past each other. Mitosis, telophase. Telophase is the fifth and final stage of mitosis, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells, also reforming nuclear envelope. A copy of each chromosome has migrated to each pole and pulled opposite of side cell. The chromosomes are surrounded by a nuclear membrane that forms at each pole while the cell is pinched in the middle for animals. Cyto. Cyto means cell. Kinesis means movement. Mitosis, cytokinesis. Cytokinesis is the process in which the cell actually divides into two. The nuclei are already at opposite poles of the cell. Cytoplasm separates and the cell pinches in the middle. The cytokinesis starts during nuclear division phase called anaphase and continues through telophase. Also, the cells are diploid, meaning they have pairs of chromosomes, as shown in the picture down below. Mitosis in animals and plants. In prophase, animal cells have centroids that are organized spindles, while plant cells do not. Plants also have a pre-prophase band, which consists of actin and microtubules that forms in the place where the cell wall will appear. The main difference between animal cell mitosis and plant cell mitosis is that in the animal cell undergoes a cell furrowing while plant cells do not because of the rigid cell wall. The major difference is that the last phase of mitosis that is telophase. Cytokinesis also occurs differently in both cells. Thank you very much.